All right, we can go ahead and get started. Thank you for that great music to start my presentation. I'm going to just share with you FaceBase as a data resource to support dental and craniofacial research. And also, I give you a case study of our recent work on craniosynostosis. So FaceBase was established in 2009 with the support from NIDCR, that's the National Institute of Dental and Craniofacial Research, as part of the NIH. The goal is to serve as a trusted online data resource for craniofacial and dental researchers worldwide. At that time, the director for NIDCR, Larry Tabak, put forward the charge for us was basically to provide this very comprehensive resource to support advancing research in craniofacial development and malformations, and also to promote collaboration among colleagues who work on craniofacial birth defects, and also integrate genomic and also phenotype data from multiple species. And the last point, which is really much more relevant now, is about data reproducibility, as I will share with you why this is so important to have a trusted data resource that we can all count on for craniofacial research. So maybe we can play the video. So FaceBase really serves as a one-stop shop for craniofacial researchers.
Although about half of all birth defects involve the face and skull, scientists remain unclear about why most occur. A comprehensive and systematic understanding is needed of how the faces of healthy children develop and what goes awry to cause common malformations. With today's improved technologies, researchers can generate many gigabytes of information in a single experiment on hundreds of genes that are involved in the process. FaceBase was created to integrate this information into a meaningful whole, while adding new data to form a more comprehensive picture of this complex developmental process.
Research in craniofacial dysmorphia could be accelerated and harder problems solved if researchers were able to collect and share their data and make it easier to find data that other researchers had produced.
In the past, research data was either not shared or organized poorly, with thumb drives lost in the abyss of filing boxes by graduate researchers long gone before publication. Some journals have ad hoc repositories, but are neither standardized nor connected with other studies. FaceBase is a comprehensive data repository that systematizes how data is presented. Basic curation and an underlying platform allow intuitive data entry with a wealth of metadata that add context and connections with other studies. FaceBase has become the standard in the field, adhering to fair principles for best practices in sharing and reuse of data for the craniofacial research community. Findable, accessible, interoperable, reusable. FaceBase datasets are validated and curated before release. Most datasets have multiple replicates to ensure data quality and reproducibility. FaceBase serves as a springboard for hypothesis-driven research, providing access to data far beyond what any one lab could generate by itself.
By sharing data at FaceBase, you have contributed to a platform where we can cross-reference each other's data and validate each other's data. You can use this platform at FaceBase as an opportunity to start future collaboration by working with others that will enable us to make discoveries that we could not do by working alone.
Submitting to FaceBase helps satisfy data sharing requirements from funding agencies like the NIH and from journals. Improve the reproducibility of your results by sharing well-documented reusable data. Increase the dissemination and impact of your research with full attribution, DOIs, digital object identifiers, data citation guidelines, and citation metrics.
So we've invested a lot of effort in understanding the sociology of the research community and the craniofacial researchers, and ensuring that control and dissemination and credit of the data is done in ways that will make them comfortable.
Over the years, we at FaceBase have developed a standard pipeline for data submission. We have streamlined the process and then developed training tools and also very standard protocol how to submit their data to FaceBase.
As researchers continue submitting data, FaceBase will continue to grow and become even more invaluable to the research community.
The technology that we're creating and advancing in FaceBase is being used and informs research in many other domains. And so what we learned from FaceBase has broad application to the advancement of biomedical science in general.
Someday in the future, that we will be able to connect all these data resources for people who work in any parts of the human body, and then make them into one comprehensive and also ever-growing data atlas.
FaceBase is really an exemplar of how science should be done in the future.
All right, so currently, if you look at the FaceBase, we have over 1,000 data sets, and that includes different model organisms. Most of the data are related to usage of transgenic animal models, such as the mouse model. We also have human data, like a 3D facial variation, and GWAS data, and also data from zebrafish, chimpanzees, and also chick data. So here is a snapshot of the last 6 months, how many people have visited FaceBase, and then how many people have downloaded data from FaceBase for their use in the research. So if you look at the landscape of how we share data in today's environment, you can basically sort of look at these data sites, put them into two different groups. One is kind of a domain specific, where you have highly detailed descriptive information about the data sets, such as geo, ZFIN for zebrafish. So we all use these data sources. But the focus is quite narrow, and then also the cost for curation is pretty high. And then you have these very generalized data resource. So where all types of data are being shared, and there are less sort of a quality control. And what we envision is FaceBase can serve as this sort of an intermediate, where we can be very flexible to adapt to the new species of people are using for their research, and also have a self-serve data curation to guide the scientists to produce quality data. And I will illustrate how that works. So if you, I don't know if you had a chance to read a publication a few years ago published by, at that time, the director of NIH, Francis Collins and Larry Tabak. And they did a study. Basically, the conclusion was only about 10% of the published data that when people try to reproduce it, they can actually, they are reproducible. So the rest of them, either because of a lack of a description of how the data was collected, or the experiment was done, or lack of sharing of the data. So make these data reproducing process very difficult. So by sharing data through FaceBase, we'll be able to improve the data quality, and also following this principle where you can increase the participation rate of the data sharing, reduce the effort in finding and cleaning and integrating data into a platform where you can find the reliable data to share. And the reproducibility is also improved by sharing data through FaceBase. So this so-called FAIR principle, findable, accessible, interoperable, and reusable, is the new standard now for data sharing in scientific research. So the key for enabling the FAIR data interaction is by having a unified data model and start with a standard vocabulary. So we have a standard vocabulary, a FaceBase, no matter what kind of data you have, whether it's human data or animal study data. And then you have to use that in order to submit the data to FaceBase. And then it's a very user-friendly interface where you can look at the 3D imaging of certain like gene expression pattern, and then or enhancer analysis. And then also this gives the ability for you to do online visualization in a 3D setting. So the data you share at FaceBase will become immediately available. So every data you submit to FaceBase will get a digital object identifier right away. Let's say if you have a set of data that you are about to submit to a journal for publication. So that can be embargoed at FaceBase, and then the reviewers for the journal will receive a token, and then they will have access to the data you have submitted to FaceBase during the review process. And that token actually is randomized, so nobody can tell who is serving as a reviewer for your paper. So it's very much confidential. Once the paper is accepted, and then FaceBase will work with you to release the data once your manuscript is published for everyone to see. So all of this can be controlled at FaceBase. And so why you wanted to submit data to FaceBase? Because we have a very broad collection of data, animal models, human subjects, imaging, omics, and other experiment types. And the commonality only focused on dental and craniofacial research. And also the data is in high quality. So the reviewers or other users who come to FaceBase, they will find your data trustworthy, then can be cited over and over again. So that really help to support the reproducibility. And also, NIH now requires every funded project to have a data sharing and management plan, meaning that the data you produce must be shared with the public. Because NIH recently did a study and learned that only about 5% of the data that NIH has paid for is actually shared in the public domain. The other 95% are sitting in somebody's lab. So that doesn't really serve anyone, right? Because if the funding agency has paid for the generation of that data must be shared with the public. So this is another very important point for FaceBase to serve in this, to support the data sharing and management plan. So how do you start with a data submission? Basically, you have a brief overview of your data and reviewed by the FaceBase steering committee. So within about 2 weeks, and we will give you a feedback whether this data is ready to be uploaded to FaceBase. And there's an online tutorial to help anyone who wants to submit data to FaceBase. And then the data submitter will describe the experiments and the biosamples using standard vocabulary. So that basically is curated from the beginning, and upload the data, and then we take over to do quality control. So we have domain experts to make sure the data submitted to FaceBase actually fits with all the requirements. And then the digital identifier is generated, so now the data becomes shareable. Anyone can cite that. And then the release of the data, as I mentioned, is really up to you when you want it to be released to the public domain. So so maybe I should give you a little bit of background how the FaceBase started. When FaceBase was first funded in 2009, it used a spoke and a hub model. NIH basically went out and solicited applications. Then they funded a hub where all the data is supposed to go. But they also selected 10 spoke projects. Those 10 spoke projects were required to send data to FaceBase. We were getting reasonable data submission from the spoke projects, but then starting from 5 years ago, NIH decided to switch to a hub-only model. But instead, they required all NIDCR-funded projects related to craniofacial research to submit their data to FaceBase. Because of this switch, we have seen a substantial growth in data submission. Basically, now every R1 funded by NIDCR has to submit their data to FaceBase. And this has really led to many new development. And then one of the, just to also illustrate how the FaceBase platform is able to integrate new data types. Recently, NIDCR funded a project called the Enamel Base. So this Enamel Base is hosted now at FaceBase in terms of the data sharing support. And within this consortium, there are about 20 different mouse mutant models generated to support a study of amelogenesis imperfecta. And they also have a in vitro bioreactor with a 3D culture system to support enamel research, different protocols, different data types. So this is a very separate project funded by NIDCR. But NIDCR made a decision to integrate all the data generated from that project into FaceBase. And we were able to create a platform that connected with FaceBase in a very seamless manner. Now, we are in the process of developing AI and machine-based learning. So one of the projects we have we will have a demo very soon, is to use basically machine learning to look at all the genetic mouse models with normal and abnormal skull development. And then so the machine will be able to give you an analysis very quickly to see what are the malformations that you will see in your animal model. And similarly, we're doing that with the human 3D analysis, so that will become available at FaceBase very soon. So some of the new initiatives that we are working on dental carie data, and so that's based on the population, the Appalachian population. The study was conducted at University of Pittsburgh and University of North Carolina. The craniosynostosis model, we have different centers within the US that are submitting data to FaceBase. Microsomia imaging and genetic data contributed by Seattle Children's Hospital and Yale University, and skull maxillofacial imaging studies focusing on malocclusion. So this is based from the intramural program at NIDCR. Thank you.
FaceBase provides various tools to visualize imaging data. Craniofacial data includes not only genetic and genomic data, but also visualizations of the soft and hard tissues of the face, skull, and teeth. Some data may also be useful for non-craniofacial research. For example, our high-resolution CT data could support the study of the brain. In this video, we'll look at 3 main types of image viewers, as well as other imaging resources available. Two-dimensional images in the FaceBase repository are commonly histological. stains to show regions where a gene is expressed. Our pipeline accepts the raw image file from a microscope and converts it to a high-resolution image that can be viewed in a browser page. Thus, the viewer acts like an online digital microscope, allowing the user to zoom in for a deep view. The viewer also supports annotations. Contributors can use editing tools to mark and label colored boundaries around specific anatomical regions. To download a screenshot of the current view, click Take a Screenshot. An accurate scale is included with the image, which is crucial for researchers and ideal for use in publications and presentations. Some imaging data is created in 3 dimensions, which is useful in understanding the shape and size of specific anatomical regions of the face and skull, for example, when comparing control specimen with mutant specimen. When a 3D image is contributed, FaceBase provides a downsampled 3D preview inside your browser window. Use your mouse to turn the 3D image in all directions and to zoom in and out. Use the right panels to step through slices of the interior through different views. Use the volume panel to adjust the levels or brightness of the planes to virtually examine craniofacial structures from a variety of sources, all within your browser. If a contributor provides surface mesh data, which depicts surfaces of interest, our surface mesh viewer can create a jigsaw puzzle effect, where each surface mesh object is given its own color and channel, which can be toggled on and off. Surface meshes may be annotated with anatomical landmarks, demarcating specific locations on a specimen. Distance between landmarks can also be calculated. This educational tool is especially powerful for new researchers to learn anatomical regions. Outside of the data browser, FaceBase offers many other atlases and resources, including landmark datasets with 3D human facial scans of a variety of populations. These scans provide comprehensive coverage of the variety of characteristics in the human face, as well as crucial measurements that can be used for applications such as designing respirators. Demographics include thousands of subjects over a range of ages, ethnicities, and some phenotypes. The 3D Facial Norms Database is a web-based resource that provides the research and clinical community with high-quality craniofacial anthropometric normative data based on digital stereophotogrammetry, a method of 3D surface imaging ideally suited for capturing human facial surface morphology. The 3D Facial Norms Database allows users to interact with data via an intuitive graphical interface and, given proper credentials, to gain access to individual-level data, allowing users to perform their own analyses. FishFace is a visual atlas of zebrafish craniofacial development that provides an interactive resource to understand not only the cellular details, but also complex 3D anatomical relationships of developing elements in the craniofacial skeleton of the zebrafish. Three interactive, three-dimensional zebrafish craniofacial atlases of anatomy and wild-type adult zebrafish classify structures of the zebrafish skull, explore the embryological origins of the bones of the skull, and emphasize the similarities between fish and mammalian skulls. The Mouse Anatomy Atlas shows color-coded images and illustrations of craniofacial regions for a wild-type mouse across early stages of development, embryonic day 9 through neonatal P0. The anatomical navigation feature, powered by our Surface Mesh Viewer, allows users to toggle different regions on and off, and click on an anatomical term to see any available related data in FaceBase. Available models at this time include mouse and zebrafish. FaceBase continues to improve and expand options for visualizing craniofacial data and accepting cutting-edge imaging formats to improve the user's experience.
So our goal is really to build this into a digital atlas. So you can do surface analysis, you can do detailed imaging of bone, soft tissue, and then you can actually go into finally to the single cell level tied with a single cell RNA-seq analysis. So it will allow you to look at gene expression in different regions of the craniofacial complex. So the other feature that you have seen is basically you can rotate these in different dimensions, and then allows you to, the platform will allow you to actually go into imaging, go into gene expression, go into looking at an enhancer analysis of a particular gene. So you can perform all type of studies.
FaceBase includes a large variety of data types that indicate where and when certain genes are expressed in various animal and human models. These data are sometimes collectively referred to as omics data, including genomics, transcriptomics, and epigenomics data. FaceBase designed its data model for reproducible omics experiments and integration of an omics pipeline within the FaceBase hub. Controlled data from wild-type animals and healthy human subjects in FaceBase serve as a rich resource for researchers as they compare their experimental data to controls. Genomic data maps an organism's entire DNA, illuminating genetic sequences, mutations, variation, and evolutionary relationships. Transcriptomic data is useful for elucidating changes in gene expression under various conditions, such as time of development in model organisms and disease models. Epigenomic data uncovers changes in gene activity not tied to DNA sequence that impact processes such as development, aging, and disease. FaceBase provides a convenient platform for downloading omics data that saves the user's time and improves data reproducibility. FaceBase is home to various types of sequencing data and other omics data types. Beyond filtering data by species, anatomy, age, and other common attributes, users can further filter omics data by experiment type, genetic background, gene, and genotype. Some examples of experiment type attributes relevant to omics data include bulk and single-cell RNA-seq assays, ChIP-seq, ATAC-seq, genome-wide association studies, whole exome sequencing, and gene expression summaries. The FaceBase platform integrates visualization tools for omics data via standardized data processing pipelines that show gene expression and cell clustering. FaceBase also hosts contributed GWAS data that show genomic coordinates for orofacial clefts, facial variation, and dental disorders. The integrated visualization from the UCSC genome browser in the record page can help researchers identify genomic data-based pair resolution. Collections of annotated datasets, referred to as tracks, provide researchers with information such as gene expression data, mapping of DNA repeat elements, and disease association data. FaceBase allows contributors to upload their sequencing data and receive processed visualization of their data. Single-cell RNA-seq data from FaceBase datasets can be visualized by the UCSC cell browser in UMAP or PCA format. The cell clusterings and gene expression patterns can be easily identified. Contributors can take advantage of FaceBase pipelines that simplify the technical work of getting data visualized. They just need to upload their data in supportive formats, and FaceBase pipelines will generate interactive visualizations for gene annotation tracks and single-cell sequencing data. The Human Genomics Analysis Interface was developed to allow users to visualize results of human craniofacial genomics projects in aggregate. HGAI is home to several craniofacial genetic projects, including studies of orofacial clefts, facial variation, and dental disorders. HGAI integrates expression data from animal models with human genomic data. For this particular orofacial clefting project, University of Michigan's LocusZoom provides users with an interactive interface to identify the strongly associated loci for significant genetic variants. LocusZoom also provides users with the ability to go to specific base pair regions on the chromosome, search for regions by gene name, and identify specific characteristics of each gene variant of interest. Beyond browsing and visualizations, advanced users can programmatically access datasets for their own analysis or bioinformatics pipelines. Our export feature is structured to export data as machine-readable and may be used for external pipelines. A user can get all of the data for one or many datasets, or for an entire project. Downloading is efficiently handled, and if interrupted, we'll retry the download and ensure integrity of data when it is downloaded. The FaceBase platform provides REST and Python interfaces, which can be used to provide standardized processing of datasets. For example, for secondary analyses. FaceBase continues to evolve its offerings of omics data in its efforts to build a community together for craniofacial, dental, and oral researchers worldwide.
So what I just hopefully I had the opportunity to share with you what FaceBase is about and then what it can do. And I also wanted to acknowledge my team of all of these people have worked together for the past 15 years to build FaceBase. Carl Kesselman is a data specialist, computer scientist at USC. We have been co-directing the FaceBase program, and then this is the list of our teams, and we are supported by NIDCR, and so really that has been such a great development for data sharing. So now, in the next 15 minutes or so, I wanted to just to give you an example of what my lab is working on related to cranial synostosis. So I think one of the important things, because I understand there are a lot of clinicians here, and so our study recently has shown that the skull and the brain, the functional sort of a connection is linked by this previously not well described lymphatic system, which really plays a very important role in brain function. So let me share with you some of the study that we have been doing. As you know, cranial synostosis come in different forms, depends on which, depends on which suture is fused prematurely. So here is a sagittal suture fusion, metopic suture, and then bilateral coronal suture fusion. And what we're going to focus on today is this TWIS1 mutation that is well known that can cause cranial synostosis in humans. And then what has not been well studied in the animal model, at least, is whether these TWIS1 mutation or FGF receptor mutation can also cause neurocognitive dysfunction. So that's what we have done in our study. As you know, in the human skull, there are 22 bones, right? And then they help to make the human skull, with the exception of our temporomandibular joint, which is articulating joint. All other bony elements are connected by sutures. So the suture provides a connection, serve as a minor movement for the joint, and also accommodate brain growth, and then serve as a shock resorption. What is less appreciated is if you look at a cross-section, here's the calvareal bone, and here are the meninges, the dura, the arachnoid, and the pia mater, and then the brain. So within this meninge, I will highlight the lymphatic system, how that helped to mediate the waste clearance, and how's that influenced by the stem cells in the cranial suture. So this, what I will share with you, sort of the two highlights of today's, the second part of the presentation. Basically, what I will share with you is if you lose these stem cells within the cranial suture prematurely, that will lead to a cranial synostosis. And then now we can actually use a biodegradable scaffold combined with the stem cells to regenerate a cranial suture. That can correct the skull deformity and also improve the neurocognitive function in the animal model. The second part is how the skull meningeal lymphatic and brain communication is orchestrated by MSC-mediated VGFC signaling. So this has been a collaboration with the neuroscientists and also craniofacial surgeons at USC. Quite a few years ago, this was a paper published from our lab. We have shown that within the adult mouse suture, there are these GLI1 positive stem cells. And then if you activate the reporter, and then you follow these blue labeled cells, after a month, you can see they have migrated into the skull, help to support bone turnover. And then if you want to test the function, how important these GLI1 positive cells are, and then you can generate a model, then eliminating these GLI1 positive cells, basically killing them. And once that happens, you can see every single suture in the skull has fused. So this clearly shows the GLI1 positive cells are important. So our model is basically the GLI1 positive cells within the suture migrate into the periosteum or into the dura and then help to support the calvareal bone expansion and bone turnover. So naturally, we would hypothesize that in a mutant animal model like TWIS1, which mimics the human condition with a suture fusion, perhaps we would see a diminished number of GLI1 positive cells prior to suture fusion. That's what we tested. So using the TWIS1 heterozygous mutant model, in which the coronal suture will fuse either unilaterally or bilaterally, and we looked at in their coronal suture, indeed, they have a reduced GLI1 positive cells. So from that, then we wanted to actually ask the question, does the TWIS1 heterozygous mutant mouse model actually represent the human condition in which they also, these patients, in the severe cases, they also have learning and neurocognitive dysfunction. So the way we tested that is basically we stick a probe into the skull and then measured the intracranial pressure. As you can see, the TWIS1 mutant has an elevated intracranial pressure in comparison to the control. And if you do a cognitive test, such as putting a mouse in a chamber and then place a new object, so see how well this mouse will try to interact with this new object. The normal mouse will naturally spend more time. As you can see, there are more lines on this side. But in the TWIS1 mutant, they fail to do so. And then similarly with social behavior. So this has been shown. So this study was published in 2021, basically showing that the TWIS1 mutant model actually mimics the human condition with these neurocognitive defects. Currently, when a patient come in with these suture fusion, they undergo these very extensive surgical procedure to correct the skull deformity. And we wanted to ask the question whether we can use a biodegradable material combined with the GLI1 positive stem cells to regenerate a suture so the skull bone can grow along with this new suture. That's what we did. And by doing that, in these 3 different groups, here's the control. Basically, we remove the fused suture, put in nothing. And then, or we just place the biomaterial, modified gelma. Or we use modified gelma combined with stem cells. Only this group, as you can see, after 6 months or a year, we get this beautiful regeneration of a coronal suture with overlapping frontal and the parietal bone. So naturally, you wanted to ask the question, are all these stem cells you put in place are still there, or they actually help to activate some of the endogenous cells to sustain this new suture? And the answer is the latter. So basically, by placing the stem cells, these are the red cells, TD tomato positive, and they were able to create this new environment and then to attract the cells from the dura in order to continue to support the patency of this newly formed suture. If we place a membrane between the calvareal bone and the surface of the dura, blocking the migration of these cells, and we can no longer regenerate a suture. So by regenerating a suture in the TWIS1 mutant model, we were able to improve the skull shape. As you can see, the green wire in comparison to the red, which is from the mutant. You can do these 3D analysis. And also, we were able to improve the neurocognitive function in the TWIS1 mutant model with a regenerated suture. So this study was published in 2021. And then, so at that time, we were very excited to see the regeneration of a suture and then can lead to this improved neurocognitive function. But we did not understand how this improved neurocognitive function took place after the suture regeneration. So this led to the second part of the study, in which we wanted to look at what are these adjacent tissue next to the calvareal bone. What kind of role these blood vessel lymphatics play in mediating the function within the brain. So if you follow the recent literature in the last 10 years, there have been a lot of study describing the lymphatic system that residing within the meninges. So when you look at the 3 layers of the meninges, the dura, where it has the outer and inner layer, at the midline here you can see the superior sagittal sinus, where you can see the rythmoid granulation that helps to drain the cerebrospinal fluid into the superior sagittal sinus. Within this space, the subarachnoid space, you will see blood vessels, cerebral artery, and vein, and also part of the lymphatic system. This lymphatic system is known to help to clear the brain waste, and then immunosurveillance, and then also serve as a buffering for the mechanical force within the skull. And disruption of the fluid through the lymphatic system has been linked to the development of such as Alzheimer's disease later on in life. And so what we have learned in the study is basically not only there's the cerebrospinal fluid circulation, but also the lymphatic system will drain into the deep cervical lymph nodes. And then disruption of this drainage can lead to malfunction in the brain. How do we test that? So in order for us to look at whether the TWIS1 mutant model also has a compromised lymphatic drainage, so you can actually inject this conjugated protein that will give you a color to see the distribution. You can see in the control sample the distribution of this conjugated protein in the subarachnoid space is throughout the surface of the brain, but in the TWIS1 mutant, there's this compromised distribution. And in terms of the lymphatic drainage, also you can see in the TWIS1 mutant, there's a diminished lymphatic drainage with the lymphatic vessel. And then once we regenerate the suture, you can see the lymphatic drainage has improved in the mutant model. So how important is this lymphatic system within the meninges? And by doing the following experiment in which you can inject this Visol dye into the subarachnoid space. And then upon using this photo conversion, you can actually eliminate a lymphatic system within the meninges. By doing that, basically, you can see a diminished lymphatic vessel and also a compromised lymphatic drainage. So using this approach, we have been able to show the importance of the lymphatic system within the circulation in the brain to help to clear the waste from the brain. So now, the question is, how do we go about testing the GLI1 positive cells we implanted into the regeneration of the suture, and how do these cells influence the lymphatic vessel? And the way we went about looking at this is first, look at on the surface of the dura, which comes into close contact with the suture, where you can see there are these GLI1 positive cells. They overlap with these lymphatic vessel marked by LYVE1. And so the question we asked is whether these GLI1 positive cells may secrete some factors to support the development of these lymphatic vessels. And the way we tested that is by culturing these mesenchymal stem cells and then harvest a supernatant from the cell culture, and then see what are the sort of the critical ingredients within this supernatant. And what we have learned is basically these supernatant from the MSC culture help to support the proliferation of these lymphatic cells, and then also support the migration of these lymphatic cells if you do the scratch assay, and the lymphatic cells can migrate much faster. And then just to make the long story short, what we learned is within this secreted factor, we identified VEGF-C. So the VEGF-C is this important factor which we think helps to support the migration of these lymphatic cells for migration. And then if we block the VEGF-C, and you can see these lymphatic cells can no longer migrate. The most important experiment to test that is by just overexpressing VEGF-C in the TWIS1 mutant model, we were able to restore the lymphatic system within the TWIS1 mutant and also improve the lymphatic drainage, and also that helped to improve the neurocognitive function in the TWIS1 mutant model. So this really helped to establish the importance of this lymphatic vessel in supporting the brain tissue waste clearance, therefore make the connection between the skull suture stem cells with the lymphatics and then the brain function. So this is what I've told you today. Basically, losing mesenchymal stem cells prematurely can lead to craniosynostosis. Since the TWIS1 study, we have also shown in the FGF mutant model, such as the APER and Cousin syndrome, they also have a diminished number of GLI1 positive cells. And then the TWIS1 mutant model, we have shown by replenishing the stem cells, we can regenerate the suture and then improve the neurocognitive function. And finally, the importance about the lymphatic system in mediating this interaction between the skull and the brain function. So this is my research group who have done all this work, and I'm really grateful for all their hard work. And then we are supported by NIDCR and also by USC AMI. And then this is the website of my group, chilab.usc.edu. You're welcome to visit and then contact me if you're ever in Los Angeles. I will be delighted to host your visit and then show you around and then we do have some good French restaurants you can visit. Thank you very much.
Well, thank you very much once again for being here today.
Yeah,
and
thank you.
for this very precise and extensive presentation of both your research and the network you've created. Do we have questions in the audience? Yes, Dr. Paternoster, she's a neurosurgeon in Necker-en-Formalade Hospital.
Hi, I'm Dr. Paternoster. I'm a neurosurgeon. I'm responsible of Craniofacial unit here in France, in Paris. Thank you very much for the very excited presentation. I'm, of course, really interesting about all the discussion of the lymphatic system and the influence on craniofacial. Probably you know that in hydrocephalus study in the last year, all the aspect of the lymphatic system and the involvement of lymphatic system is becoming more and more something that we are looking for. So my first question is that, have you seen any correlation in the mutation group with the lymphatic abnormalities on hydrocephalus in this patient, or in some way can increase the risk of hydrocephalus in this group of mutation group? And the other question is that we have the impression, especially in the Cruzon and Pfeiffer patient, that all the venous problem, you know, the stenosis and the development of collateral system can be not simply related to the closure of the bone, but more something related probably to mesenchymal abnormality or mesenchymal problem. So I don't know if also you have noticed this correlation, this influence also on the venous part of the problem, especially to explain a little bit better the increase of intracranial hypertension in this group of patients.
Yeah, so thank you for all your questions. And so regarding the first question, the lymphatic system in patients. So far, what in collaboration with Andrew Wilkie's group, who have actually generously provided us with some patient samples, and they were actually blinded, and then so we looked at, at that time, we only looked at the GLI1 positive cells, the number of the stem cell. GLI1 is, you know, we use as a marker. It's an imperfect marker, but what we did see is in both APERS and also Cruzon and the Cedar-Chosen syndrome patients, they have a diminished number of GLI1 positive cells. And we have not looked at the lymphatic system in these patients. That's something we plan to do. And regarding the second part of your question about, you know, the changes in the skull, that's not sufficient to explain all the phenotypes that we see. I absolutely agree with you. What we have seen is a very general change in the dura in the TWIS1 and also in the FGF receptor mutant model, the animal model we have looked at. They have a diminished number of GLI1 positive cells throughout the surface of the dura. The other part I also wanted to, these are all unpublished studies that we have been working on. If you look at the skull bone, and then, you know, naturally, we always wanted to ask the question, you know, why is the coronal suture or the sagittal suture affected, but now not lambdoidal suture? It's so rare to see lambdoidal suture fusion, right? And then, but when you look at the GLI1 positive cells, they're everywhere. So this has led to some of the studies now we're looking at not only the suture, but also the local environment. What I can tell you is, if you look at the bony structure, like the frontal and the parietal bone, there are very few GLI1 positive cells in the bone marrow of the frontal and the parietal bone, but in the occipital bone, there are tons of GLI1 positive cells in the bone marrow. And so that difference, I think, can be sort of something to explore further. And also, in terms of the difference between coronal and sagittal suture, the reference or the connection with the superior sagittal sinus versus, you know, just the dura surface, those are the things we're looking into, but I don't really have an answer for you. But I think the neighboring tissue will play a very important role in our understanding about why a particular suture always gets affected, not the other ones. Yeah.
Thank you very much. Other questions? Another questions in the audience? Well, we'll save. Also, I have perhaps just a short one. Yes, about the network you've created. I guess you have already addressed the issue, but what happens when the paper is submitted, accepted? I understood that the reviewers can get online the pictures, and what happens when the pictures become the property of the journal that publishes the work in? Does it stay on your website also, or on the website of the network? Well,
yeah.
Yes.
So
how do you share the copyright?
Yeah. So the question is whether how do we deal with this once the paper is published? All the figures become the property of that particular journal. So FaceBase will still maintain all of the raw data that you use to generate those figures. So those raw data will still be available for people to use. We don't really publish or put the figures in your paper on FaceBase. So it is only the raw data that you use to generate your figure. Yeah.
Okay.
Yeah.
Thank you.
So those will be always be available for people to share and to use.